Animal cell culture, MCQs. Which scientist is known as the father of animal cell culture? A. Wilhelm Roux. B. Ross G. Harrison. C. Alexis Carrel. D. Harry Eagle. Answer, B. Explanation, Ross Granville Harrison, 1907, first demonstrated long-term growth of animal cells in vitro using the hanging drop technique. The first successful long-term animal tissue culture was performed using A. Mouse embryo cells. B. Frog embryo nerve cells. C. Chick heart tissue. D. Human skin fibroblasts. Answer, B. Explanation, Harrison used frog embryo spinal cord cells to demonstrate nerve fiber outgrowth in vitro. HeLa cells are derived from which type of tissue? A. Human liver. B. Human cervical cancer. C. Mouse kidney. D. Human lung epithelium. Answer, B. Explanation, HeLa cells were derived from Henrietta Lacks cervical carcinoma cells in 1951. Which of the following media is richest in nutrients and commonly used for mammalian cell culture? A. PBS. B. DMEM. C. LB broth. D. Peptone water. Answer, B. Explanation, DMEM, Delbecco's modified Eagle medium, contains high glucose and amino acids suitable for fast-growing mammalian cells. Which amino acid in culture media is unstable and must be added fresh? A. L-arginine. B. L-glutamine. C. L-tyrosine. D. L-methionine. Answer, B. Explanation, L-glutamine decomposes into toxic ammonia, hence it is freshly added to media before use. The main buffering system in CO2 incubators relies on A. HEPS buffer. B. Sodium bicarbonate, CO2 equilibrium. C. Tries buffer. D. Phosphate buffer. Answer, B. Explanation, CO2 reacts with NaHCO3 in the medium to maintain physiological pH, 7.2 to 7.4. The main energy source in most animal cell culture media is A. Sucrose. B. Glucose. C. Lactose. D. Glycerol. Answer, B. Explanation, glucose provides ATP via glycolysis and oxidative phosphorylation. Phenol red in cell culture medium is used as A. Nutrient. B. Antibiotic. C. pH indicator. D. Growth factor. Answer, C. Explanation, phenol red changes color based on pH, red equals neutral, yellow equals acidic, purple equals basic. Which of the following is not an example of a continuous cell line? A. HeLa. B. CHA. C. Vero. D. Human fibroblast primary culture. Answer, D. Explanation, primary cultures have a finite lifespan, unlike continuous, immortal, cell lines. The first immortal human cell line established was A. Vero. B. HeLa. C. BHK21. D. HEC293. Answer, B. Explanation, HeLa cells were the first immortal human cell line established in 1951. Which of the following is used to detach adherent cells during subculturing? A. Collagenase. B. Trypsin-EDTA. C. Pepsin. D. Lipase. Answer, B. Explanation, trypsin-EDTA breaks peptide bonds and chelates Ca superscript 2 to release adherent cells. Cryopreservation of cells typically uses which cryoprotectant? A. Glycerol 10%. B. DMSO 10%. C. Ethanol 5%. D. Methanol 20%. Answer, B. Explanation, DMSO prevents ice crystal formation and protects cell membranes during freezing. Which of these is not a component of typical animal cell culture media? A. Amino acids. B. Inorganic salts. C. DNA. D. Vitamins. Answer, C. Explanation, DNA is not part of media, cells synthesize their own nucleic acids. Which serum component helps in cell attachment to surfaces? A. Albumin. B. Transferrin. C. Fibronectin. D. Insulin. Answer, C. Explanation, fibronectin promotes cell adhesion and spreading on culture surfaces. What is the function of transferrin in serum supplemented media? A. Glucose transport. B. Iron transport. C. Calcium buffering. D. pH control. Answer, B. Explanation, transferrin carries phase superscript 3 required for enzymes and proliferation. Which equipment maintains sterile airflow in a culture environment? A. CO2 incubator. B. Centrifuge. C. Laminar airflow hood. D. Hot air oven. Answer, C. Explanation, laminar hoods provide HEPA-filtered sterile airflow for aseptic handling. Optimal temperature for most mammalian cell culture is A. 20 degrees Celsius. B. 25 degrees Celsius. C. 30 degrees Celsius. D. 37 degrees Celsius. Answer, D. Explanation, 37 degrees Celsius mimics normal human body temperature. Vero cells are derived from A. Mouse embryo. B. Monkey kidney. C. Human lung. D. Hamster ovary. Answer, B. Explanation, Vero cell line comes from African green monkey kidney epithelial cells. CHO cells are widely used for production of A. Antibiotics. B. Recombinant proteins. C. Viral DNA. D. DNA vaccines only. Answer, B. Explanation, CHO, Chinese hamster ovary, cells are ideal for large-scale recombinant protein production. Which enzyme is responsible for enzymatic dissociation of tissues? A. Amylase. B. Collagenase. C. Maltase. D. Catalase. Answer, B. Explanation, collagenase digests extracellular collagen, freeing individual cells. Which of the following best defines passaging? A. Freezing cells. B. Transferring cells to new vessels. C. Adding antibiotics. D. Measuring cell viability. Answer, B. Explanation, passaging, subculturing, is transferring cells to new vessels for continued growth. The most common contamination in cell cultures is A. Fungi. B. Bacteria. C. Mycoplasma. D. Protozoa. Answer, C. Explanation, mycoplasma are small bacteria lacking cell walls, they are hard to detect and cause chronic contamination. Which chemical disinfectant is used to clean culture surfaces before work? A. Chloroform. B. 70% ethanol. C. Benzene. D. Acetone. Answer, B. Explanation, 70% ethanol denatures proteins and is ideal for surface disinfection. In cryopreservation, cells must be frozen at approximately A. 10 degrees C/min. B. 1 degree C/min. C. 0.1 degree C/min. D. 5 degree C/min. Answer, B. Explanation, slow cooling, 1 degree C/min, prevents ice crystal formation and increases survival. Which is not an advantage of animal cell culture? A. Controlled environment. B. Ethical alternative to animals. C. Fast growth rate compared to microbes. D. Study of disease mechanisms. Answer, C. Explanation, animal cells grow slower than microbial cells. Which of the following is a serum-free media supplement? A. FBS. B. Transferrin. C. Blood plasma. D. Albumin. Answer, B. Explanation, serum-free media include defined supplements like transferrin, insulin, and growth factors. Who developed minimum essential medium, MEM? A. Harry Eagle. B. Alexis Carrel. C. Jonas Salk. D. Henrietta Lacks. Answer, A. Explanation, Harry Eagle formulated MEM in 1955 for mammalian cell culture. The phenomenon of contact inhibition is lost in A. Normal cells. B. Cancer cells. C. Primary fibroblasts. D. Stem cells. Answer, B. Explanation, cancer cells continue dividing even when crowded, normal cells stop dividing due to contact inhibition. HeLa cells were primarily used to test which major vaccine? A. Rabies. B. Polio. C. Hepatitis B. D. Influenza. Answer, B. Explanation, HeLa cells were instrumental in developing the first polio vaccine by Jonas Salk. Which vitamin acts as an antioxidant in cell culture media? A. Vitamin B2. B. Vitamin A. C. Vitamin C. D. Vitamin K. Answer, C. Explanation, ascorbic acid, vitamin C, neutralizes free radicals and supports collagen synthesis. During cell counting, trypan blue is used for A. Measuring protein concentration. B. Distinguishing live and dead cells. C. Counting only dividing cells. D. Estimating DNA content. Answer, B. Explanation, trypan blue stains only dead cells, membrane permeable, allowing viability estimation. In animal cell culture, which gas mixture is typically used in CO2 incubators? A. 95% O2 plus 5% CO2. B. 5% CO2 plus 95% air. C. 10% CO2 plus 90% N2. D. 100% CO2. Answer, B. Explanation, 5% CO2 maintains bicarbonate buffering, and 95% air ensures oxygen availability. What is the function of sodium pyruvate in culture media? A. Nitrogen source. B. Energy supplement. C. Buffering agent. D. Antibiotic. Answer, B. Explanation, sodium pyruvate provides an alternative energy source for cell metabolism. In tissue culture, the anchorage-dependent cells grow. A. In suspension. B. On solid surfaces. C. Inside semi-solid gels. D. Only in serum-free media. Answer, B. Explanation, most animal cells, example fibroblasts, need attachment to a surface for growth and division. The process of transferring a small number of cells to a new flask for further growth is called A. Seeding. B. Subculturing. C. Plating. D. Harvesting. Answer, B. Explanation, subculturing maintains cell viability by avoiding overcrowding and nutrient depletion. Which of the following cell types are anchorage independent? A. Fibroblasts. B. Epithelial cells. C. Lymphocytes. D. Endothelial cells. Answer, C. Explanation, lymphocytes and hematopoietic cells grow freely in suspension. The most common method to check contamination in cell culture is A. PCR. B. Microscopy. C. Spectrophotometry. D. ELISA. Answer, B. Explanation, microscopic examination detects turbidity, floating debris, and microbial contamination. Hybridoma technology combines A. B. Lymphocytes and myeloma cells. B. Fibroblasts and stem cells. C. Neurons and glial cells. D. Macrophages and RBCs. Answer, A. Explanation, fusion of antibody-producing B cells with immortal myeloma cells produces monoclonal antibodies. In animal cell culture, antibiotics are used to A. Stimulate cell growth. B. Prevent microbial contamination. C. Increase protein expression. D. Enhance differentiation. Answer, B. Explanation, antibiotics prevent bacterial or fungal contamination but should not replace sterile technique. Which of the following is not an application of animal cell culture? A. Vaccine production. B. Genetic engineering. C. Soil fertility improvement. D. Cancer research. Answer, C. Explanation, animal cell culture is unrelated to soil biology, it's used in biomedical research. The first vaccine produced using animal cell culture techniques was A. Rabies. B. Polio. C. Smallpox. D. Measles. Answer, B. Explanation, the polio vaccine was mass-produced using monkey kidney cell cultures. Which of the following equipment is essential for maintaining aseptic conditions? A. Laminar airflow cabinet. B. Centrifuge. C. pH meter. D. Spectrophotometer. Answer, A. Explanation, the laminar hood ensures sterile airflow to prevent contamination during handling. Why is serum sometimes avoided in industrial cell culture systems? A. It is inexpensive. B. It introduces variability and contamination risk. C. It contains antibiotics. D. It prevents cell attachment. Answer, B. Explanation, serum-free media are preferred for consistency and safety in biopharmaceutical production. Which of the following is an immortalized kidney cell line used for vaccine production? A. CHA. B. HeLa. C. Vero. D. BHK21. Answer, C. Explanation, Vero cells from African green monkey kidney are used in polio, rabies, and COVID-19 vaccine production. Which cell line is most commonly used for recombinant protein expression? A. HeLa. B. CHA. C. HepG2. D. NIH3T3. Answer, B. Explanation, CHO, Chinese hamster ovary, cells are industry standard for therapeutic protein production. In suspension cultures, aeration and mixing are usually achieved by A. Stirred bioreactors. B. Rotating drum. C. Roller bottles. D. Static flasks. Answer, A. Explanation, stirred tank bioreactors provide controlled oxygen and nutrient distribution in suspension cultures. Which biosafety level, BSL, is typically required for handling HeLa cells? A. BSL-1. B. BSL-2. C. BSL-3. D. BSL-4. Answer, B. Explanation, HeLa cells are human-derived and require BSL-2 precautions due to potential contamination risk. Which technique is used to authenticate a cell line's identity? A. Western blot. B. STR profiling. C. ELISA. D. PCR for ribosomal RNA. Answer, B. Explanation, short tandem repeat, STR, profiling ensures that the cell line matches its genetic origin. A confluent monolayer in cell culture refers to A. Dead cells floating in medium. B. Cells covering entire surface area. C. Freshly seeded cells. D. Suspension culture only. Answer, B. Explanation, when cells cover 100% of the growth surface, the culture is said to be confluent. The importance of animal cell culture in modern biotechnology lies mainly in A. Soil fertility. B. Study of plant metabolism. C. Production of recombinant therapeutics and vaccines. D. Mining and metallurgy. Answer, C. Explanation, ACC provides controlled systems for producing monoclonal antibodies, recombinant proteins, and viral vaccines. Cryopreservation of animal cells is usually carried out using A. Ethanol. B. Glycerol. C. Dimethyl sulfoxide, DMSO. D. Acetone. Answer, C. Explanation, DMSO prevents ice crystal formation that damages cell membranes during freezing. What is the ideal temperature for long-term storage of animal cell lines in liquid nitrogen? A. 20 degrees C. B. 70 degrees C. C. 120 degrees C. D. 196 degrees C. Answer, D. Explanation, cells are stored at 196 degrees C, liquid nitrogen, to maintain viability indefinitely. The process by which normal cells become capable of indefinite division and tumor formation is called A. Transfection. B. Transformation. C. Differentiation. D. Mutation. Answer, B. Explanation, transformation refers to conversion of normal cells into immortal or cancerous ones. Which assay is most commonly used to determine cytotoxicity in cultured animal cells? A. Southern blot. B. MTT assay. C. Northern blot. D. PCR assay. Answer, B. Explanation, MTT assay measures mitochondrial activity, indicating cell viability or death after drug exposure. Which of the following bioreactor types is most suitable for large-scale animal cell culture? A. Airlift reactor. B. Stirred tank reactor. C. Packed bed reactor. D. Rotary drum reactor. Answer, B. Explanation, stirred tank bioreactors ensure controlled aeration, pH, and mixing for large-scale culture. Which statement best describes anchorage dependence? A. Cells divide only when attached to a surface. B. Cells grow freely in suspension. C. Cells divide without any attachment. D. Cells divide only at high CO2 levels. Answer, A. Explanation, many animal cells need surface attachment, via INT egrins, to initiate division and survive. What is the function of L-glutamine in animal cell culture media? A. Vitamin precursor. B. Carbon source for DNA. C. Nitrogen source and energy substrate. D. Antibiotic. Answer, C. Explanation, L-glutamine serves as a nitrogen and energy source essential for nucleic acid and protein synthesis. Mycoplasma contamination in animal cell culture can be detected by A. Gram staining. B. DAPI staining or PCR. C. SDS page. D. Western blotting. Answer, B. Explanation, Mycoplasma lacks a cell wall, detection is done using DNA-binding dyes like DAPI or PCR assays. Which of the following best defines hybridoma cells? A. Hybrid between virus and bacteria. B. Fusion of normal B cells with myeloma cells. C. Combination of fibroblast and epithelial cells. D. Hybrid of yeast and mammalian cells. Answer, B. Explanation, hybridomas are immortal cells that continuously produce specific monoclonal antibodies. Which biosafety level, BSL, is required for culturing primary human cells potentially infected with pathogenic viruses? A. BSL-1. B. BSL-2. C. BSL-3. D. BSL-4. Answer, C. Explanation, primary human tissues may contain infectious agents, thus handled under BSL-3 for safety.